Very good evening to all. I am Prabodhani from University of Vocational Technology. It is privilege for me to welcome you all on behalf of the Food Technology Association, University of Vocational Technology. This is the third session of the Industrial Experts View webinar series. The topic of the pride in the plant protein. Sorry for mistake. Very good evening to all. I am Prabodhani from University of Vocational Technology. It is privilege for me to welcome you all on behalf of the Food Technology Association, University of Vocational Technology. This is the third session of the Industrial Experts View webinar series. The topic of pride in the plant protein wave along with the food science, and we look forward to listening to how best we can improve our knowledge of what we have inherited. So, this is the time to welcome our today's guest speaker. He is a specialist in food science, research and development, graduated from University of Trichavardhanapura, and he had 4-year working experience in North Dakota State University and has a PhD in food science. Presently, he is a research and development scientist at Erdon Mills, USA. He is none other than Dr. Supun Sandaru Fernando. Dear sir, we warmly welcome for today's session. I am sure today's session will be a very interesting session, and a special reminder you all, make sure your microphones are muted throughout the session. So, now I invite you, sir, to continue this session.
Can you make sure that everyone can hear me?
Yes, sir, yes.
Okay, thank you. So good evening, everyone. So it's morning here actually, but it's my great pleasure to invited by the Association of Food Science and Technology in the University of Vocational Training to deliver this guest lecture. Before we start, I would like to share one activity with you, a link in the chat. So if everyone go to that chat box and look at the link and participate in that poll, so I can make sure that how much of awareness we have right now on the plant proteins. So it will pave the way to the presentation that I'm going to do. So if you can participate in this poll, it will be great. And also I will be keep off my video because to get rid of all the interruptions that we can have during the presentation, because my files are a little bit larger. So I will keep my presentation, my video turn off during the presentation. And when we move to the question and discussion session, I will turn on, and then we can discuss the questions you have. For each question, I will be giving you like 20 seconds. So I see many of you have participated, and thank you for your participation. Let's move on. I see most of us have a little bit of background on the plant protein.
Please excuse me, sir.
Yes, go ahead.
Sir, waiting සර්ට තමයි අපි දැන් host එක දීලා තියෙන්නේ sir. පොඩ්ඩක් sir බලනවා sir අපි waiting list එකේ කවුරු හරි අනිවාර්යෙන් තව ඉන්නවා participants ලා. පොඩ්ඩක් අපි පොඩි technical error එකක් තියෙනවා post කෙනෙක් නැති නිසා.
මම බලන්න ඒක admit කරන්න තියෙන්නේ.
ඔව් sir. ඔව්.
මම දැනට හිටපු ඔක්කොම වගේ ඇඩ්මිට් කරා. සැරින් සැරේ කට්ටිය ජොයින් වෙන හින්දා මම තව විනාඩි 5ක් වගේ දීලා ඔක්කොම ඇඩ්මිට් කරන්නද එකපාරට?
ඔව් සර්. ඔව් අපි 5යි කාලෙකම් බලමුද සර්?
හරි. ඒ වුණාම මම ෂෙයා කරන්න මගේ ප්‍රසන්ටේෂන් එක. නැත්නම් සැරින් සැරේ එනකොට මේ මට බලන්න
ඔව් ඒක තමයි අන්න එක. 6 කාලට අපි මේ කරමු.
හරි. I'm going to wait another minute to admit all the participants, and then at 6:15 from Sri Lankan time, I will be starting my presentation.
Okay, sir. Okay.
Seems like it's 6:15 in Sri Lankan time, so I'm going to start the presentation. Can somebody again confirm that everyone can hear me?
Yes, sir. Yes. Okay.
Okay, thank you. And can you see my
Yes, sir,
presentation
yes,
as
sir.
well? Okay. So good evening and thank you for joining this guest lecture on riding the plant protein wave. And again, it's my pleasure to accept the invitation from the Unitech Food Technology Association and deliver today's online lecture. So to briefly introduce who I am, my name is Supun Fernando, and I'm a R&D scientist. I have a bachelor's degree in food science and technology from University of Sri Jayawardenepura and master's and PhD in food science from North Dakota State University in US. My master's thesis and doctoral dissertation work were focused on dry bean milling and flour functionality. Before I moved to US, I worked at Kiehl's Food Products, University of Sri Jayawardenepura. Can everyone please mute your microphone so it will be easy for me to do the presentation? So before I moved to US, I worked at Kiehl's Food Products, University of Sri Jayawardenepura and Aquinas University College. During my graduate studies, I was able to do 2 internships at 2 food ingredient companies in US, and I have gained much experience in milling and plant proteins. After I graduated with my PhD, I started working as an R&D scientist at Ard Mills. To give some glimpse of Ard Mills is the largest flour milling company in North America and the second largest in the world. We feed over 100 million people every day. So wheat milling is our core business. Currently, as a R&D scientist, my main focus is on pulses and plant protein ingredients. So this is today's content. I will start with a brief introduction on what are plant proteins and why plant protein topic has become mainstream. Then I will take you through some of the plant protein sources and their manufacturing techniques. Later, I will explain why it is important to modify those native plant proteins and how to incorporate into food products accordingly to satisfy the consumers. I will try to mute everyone from my side.
Here, Maria.
There you go.
So I think now I am the only one who is speaking. Okay. So finally, so looking at the contents for today, starting from what are the plant proteins and why plant proteins are important. And then I will talk about the plant protein sources and how to manufacture those plant proteins. And what are the methods of modification for these plant proteins, because we can't always use these native plant proteins in the food products. And finally, I will go through what are the products we can make from these plant proteins. And as I will talk about one successful story and wrap up with the research gaps that we see in this plant protein arena. And then for today's lecture, I will be utilizing two of my recent peer-reviewed journal articles and a magazine article where I was a guest writer. If you like to read and learn more on this topic, let me know. So I will be happy to share these articles with anyone who is interested. Okay, so who is ready to ride the plant protein wave? Let's start with the basics. What are plant proteins? Or in general, what are proteins? Proteins are polymers of amino acids. Proteins perform a vast array of functions within organisms, providing the structure to cell and organisms, and sometimes it transports molecules from one location to another, and it catalyzes metabolic reaction, DNA replication, and it also responds to stimuli. Most importantly, proteins differ from one to another, primarily in their sequence of amino acids, which is dictated by the nucleotide sequence of their genes, and which is usually results in the protein folding into specific 3D structure, and that 3D structure will determine its activity. Each plant protein species has a unique composition of protein types. So when you look at the plant cell structure, you can see that plants also contain non-protein components like starch, fiber, and oil. There are some advantages as well as disadvantages with the plant proteins. As of disadvantages, there are formulating difficulties due to native plant protein functionality, and then some unfavorable organoleptic properties, and also some plant proteins lack some of the essential amino acids and vitamins. When we look at the advantages, they are healthy compared to animal proteins, good for planet, and it's more sustainable in protein conversion efficiency. So let's move to the next slide, where I will emphasize why there is a huge need of moving from animal proteins to plant proteins as consumers. United Nations projections have indicated that the global populace will increase to 10 billion by 2050. One of the most significant challenges faced by the world today is to ensure that the growing population has the access to adequate, sustainable, and nutritious food. This growing population will demand 30 to 50% more proteins in future. So animal proteins alone can't fulfill this requirement, and it won't be sustainable. Food production contributes more than 25% of global greenhouse gas emissions. Our current agriculture system uses 50% of habitual lands and 70% of fresh water. Most of them are accounted for animal agriculture. And also, for an example, with the coronavirus, 3 out of 4 emerging infectious diseases are zoonotic, meaning it's transmitted from animals to humans. Crowding, chronic stress, and poor health facilitate the pathogens to mutate in these animal agricultural systems. And also the current dietary patterns results in up to 5.1 million additional deaths through chronic disease mortality compared to a more plant-based diet in line with dietary guidelines. So there is a huge drive towards plant-based foods due to the growing awareness of the detrimental effects of animal meat on human health. the environment, and its inefficiency in protein conversion. Mintel, a global market research company, has done a large study on consumer preference on plant proteins, and here are some of the results. You can clearly see that the health is the main driver for eating plant-based foods. Environmental concerns are secondary. Here you can see it's 56% people are saying they are eating plant proteins to be healthier, but mostly whether it's environmental or animal concerns, they come to the second. Also, when we look at the investments on the alternative protein market, plant proteins make bulk portion of the investment compared to other alternative protein investments. On another note, alternative proteins include plant-based and fermented derived proteins and cultured proteins. So I'm not going to go into details of the other two categories, these fermentation and the cultivated proteins today. So looking at all this data with this market research and looking at the current investment, we can see that plant protein is not a niche market anymore. It's now mainstream. So what are the plant protein sources available right now? Protein sources in general can be categorized based on many criteria. It can be either plant-based or animal-derived, or it can be established, emerging, or future, or even within plant proteins, it can be cereal-based, or either it can be legume-based. When we consider different plant protein sources for our consumption or product development, there are a few characteristics that we need to pay attention. Protein concentration is how much protein we can extract from a plant source. PDCAS is related to the nutrition value. It's protein digestibility corrected amino acid profile, amino acid score, which means how much protein it's actually absorbed by the body. It doesn't make any sense, even though you have like a higher protein concentration from one plant source, but if it can't be absorbed by your body. So then we need to consider about the allergen risk. Some plant sources like soy can be allergenic to some people. And what's the commercial stage? Whether it's already established or still it's in the R&D state. Some plant sources like pea and beans, it can be less flavorful when you put it into a food product, but some are not. And then the functionality, let's say if you are going to make an ice cream out of pea protein, so what's the functionality? Can you make the foam out of that pea protein? Compared to a protein from a different plant source like sorghum protein. And what's the cost? How effective and efficiently you can extract the protein and it can be put it into a food product. And currently, what's the global crop volume? Whether it's already planting in a higher rate or still it's in a lower rate of harvesting. So this table was taken from Good Food Institute 2021 report, where it summarized the protein characteristics of different plant sources. If you look at each plant source, their proteins have different characteristics. So it can extract in high protein concentrates. Like here, you can see it in green color. That means excellent. You can extract higher protein concentrate up to 90%. And then when you look at the PDCAS, it's also a green. That means the PDCAS is high, but the allergenic, it can be somewhat allergenic to some of the people. Compared to soy protein, pea protein is now gaining some traction in the market to replace most of the soy products. But as I mentioned, so the protein concentration is not still up to 90%, and PDCAS is still low compared to soy proteins. But the good thing is it's a lower allergenic risk for people, and the commercial stage is improving, functionality is improving, and also the crop volume is improving. So likewise, we have to categorize all the plant sources and then decide, okay, when we are going to make a food product, what's the best source we can use? So, and the other thing I want to mention, not only single plant protein sources, but also blending 2 or more different plant sources can help to achieve specific product development goals. Sometimes they can synergistically work to make the structure of the food products. Sometimes they complement each other in terms of amino acid profiles. Like for an example, we do it in Sri Lanka by eating rice and lentil in our meals. And sometimes to make a favorable meal, to make the flavor tuning by mixing 2 different protein ingredients. Since there is a wide range of plant proteins, I will take legume as an example for manufacturing, modification, and product development in next few slides. As the global protein demand is expected to double in coming decades due to the increase in population, as I mentioned in the earlier, and new consumer trends, legumes or pulses have been identified as a nutritious and inexpensive alternative source of protein. Apart from their nutritional properties, legume proteins have been identified as having useful functional properties for food products, including desirable solubility, emulsifying, forming, gelling, and water and fat binding properties. The development of a more sustainable separation or fractionation method that preserves the nutritional and functional properties of legume protein is important, because now we know, okay, the legume proteins have good functional properties, but how are we going to extract this protein out of the seeds? So we have to come up with a more sustainable and a more efficient way to extract these proteins. So before we move to the manufacturing methods, first I want to give you a little introduction to the seed structure of the legumes. So this is a chickpea seed, you can see. And then in general, even though I'm just using chickpea as an example for most of the pulses, the same seed structure they have. So pulse seed has 3 main anatomical parts. Seed coat, this is the outer cover, we call it seed coat. And the cotyledon is the middle part, or where the starch and proteins are stored. And then the embryonic axis, so that's the part that is responsible for the growth of the seed into a plant in the next generation. And if we dive deep into the cellular structure, you can see that the starch granules are embedded in a protein matrix. So here you can see these are starch granules, and this is the protein matrix. And also you can see the outer fiber. So if you can't imagine how it's in the cell level, think of it as a football being entangled in a football net. So our goal is to separate these starch granules from the protein. So that way we can make 2 different streams, one for protein and one for starch. So in plant protein extraction or manufacturing, there are 2 main methods, wet method and dry method. So dry separation is energy efficient and results in high yield, where wet separation have high product purity, but at the same time it has high energy requirement. Legumes can be divided into those that have store energy as starch and those that store energy as oil. Starch-rich legumes such as pea, chickpea, and beans, they can be dispersed in water to dissolve the protein and suspend the starch granules. Subsequently, the protein is recovered as a slurry by separating the starch granules in hydrocyclones. Oil-rich legumes like soy and lupine, they are first subjected to a solvent extraction to remove the oil. The defatted flour is then suspended in water and a suspension of protein and fiber is obtained. For both starch-rich and oil-rich legumes, solubilized proteins are separated from insoluble fiber at pH of 9. So we call it alkaline extraction. Proteins then get separated from the soluble fibers by precipitating at the isoelectric point of pH of 4.5, and then subsequently the pH is readjusted to 7 to make it neutral, and a dry protein isolate is obtained by final drying step by spray drying. But the problem with this wet fractionation method is this process consumes large amount of water and chemicals and also energy. So compared to the wet fractionation, we are going to look at what's the difference between the wet fractionation and the dry fractionation. So a more sustainable alternative to obtain protein-enriched fractions from legume is dry fractionation, which uses only milling and air classification. The addition of water is not necessary here, and thus an energy-consuming drying step is not required. Another advantage is that the absence of drying step. So there's no drying step like we saw in the wet fractionation. We had to spray dry, but here we don't have a drying step, and also the absence of chemicals. And so these two steps, we don't use spray drying, we don't use chemicals. So this will protect the native functional properties of the proteins from the dry fractionation method. The major drawback in this dry fractionation is it's relatively modest enrichment, which means you can't achieve a higher purity like you would achieve in wet fractionation, because in wet fractionation, you can go up to 85%, 90% of protein, but in the dry fractionation, you can only achieve 75% or 80%. But the good thing is most of the food systems do not contain very high protein purity, but they only utilize the complex composition of the protein, starch, fiber, and other components to achieve the desired functionality. So therefore, it is not always necessary to separate ingredients into individual components with high purity at their molecular levels. Like you don't need to achieve this 90% protein concentration for most of the food products that you are going to make. You can achieve your food product by 75% of protein concentration. So furthermore, due to mild processing conditions, protein concentrates are they have the native functionality, and it's a good thing compared to the wet fractionation. And apart from these two different fractionation method, wet or dry, now you can use hybrid method. You first start with the dry fractionation until you come to the fine fraction. So this fine fraction is the protein, coarse fraction is the starch. So after you achieve this, if you want to further purify your material, you can use another step using water to purify your protein material. So we call it hybrid. So you start with the dry methods, it's only milling and air classification. We don't use water until up to this point. And later we use the water to purify more of your protein out of some of the insoluble fibers. So I would like to pay more time to the dry fractionation. So I just explained the overview in the previous slide, but I would like to dive deep into the dry fractionation method. So you start with the seeds from here, and the first step will be dehulling. So if you can remember the seed structure that I showed you in a previous slide with the chickpea, we have the outer cover. And now our goal is to remove that outer cover because most of the anti-nutrients and the unfavorable favor compounds are mostly present in the seed coat. So dehulling is the process of seed coat removal from the seed. Dehulling of legumes prior to milling and separation can affect the physical properties, chemical composition, and the techno-functional properties of your final protein fraction. Attrition type dehullers like hammer mills, which use the impact force, are suitable for dehulling legumes with loose seed coat because some grains have a loose seed coat. It is easy to remove the seed coat. Some are not. You have to abrase it very hard to remove the seed coat or the outer cover. So the abrasive type dehullers are used to remove the seed coat of seeds that are having a tightly adhered seed coat. But all in all, you can use some pre-treatments like we call it tempering. So you put water and you shake it and keep for a while. So then it will be easy to run through the dehuller to separate your seed coat. So in a normal wheat milling process, that's the first step. You get the wheat and you temper into a certain level of moisture level. And then you run it through the mill. Then it's easy to separate the bran out of the seed. So the same principle we are going to use for the legumes or pulses. You can put water, you shake it, we call it tempering, and then the seed coat will be loosened from the cotyledon. There will be a gap. So when you run it through the abrasive type mill, you can remove the seed coat. So after dehulling, we come to the milling and air classification. So this is the main part of the dry fractionation. So I'm sharing a small video clip on how the milling and the air classification happens. So this is the basic setup for the milling and the air classification. So you have a dosing unit, classifier mill, and the classifier itself. And then you have two different separate streams for the coarse product and the fine product. So the blue color arrows were the air that comes to the system, and this is the material feeding through the feeding funnel. And there's a detection system to remove the impurities like metals or any other impurities like stones. And then it will be fed into the mill. It's an impact mill where you throw the materials into the wall. So then it will be burst into small pieces. So this is the part, the impact mill. So the small particles after it get milled, it will be thrown into it will be sent into the air classifier for further classification. So when you throw the material, it will hit the wall or the plates, then it will be break into small pieces, and then it will go through the impact classifier itself to the large classifier. So from the classifier, it will select the small particles and the large particles. So the small particles will be come down to the protein. It will come down as the protein fraction. And the large particles will come down as the starch fraction. For the entire system, we are not using any water or chemicals, as you saw. We just feed the material. It breaks into small pieces using air. We carry them, and the classifier, classifier as an sieve. So if it is small enough to go through the classifier, and it will go to the protein stream. If not, it will be collected into the starch stream. So I explained to you how the starch and protein present in the cell structure. So it's like the football net, like the footballs are entangled in that net. So we are separating those footballs from the net. So footballs are starch granules, and the net is the protein matrix. So in total, the principle is we have to carefully mill our material, mill our seeds into a fine particle size, and then separate them based on their size and density. You saw that we only use the air. So what air is going to do, because we now mill into a small particle size, starch and protein have different particle size and density. Starch are largest in size, and also the density is high. So that's why when you put air to the milled material, it can easily separate protein because it's small, it's less in density, so it can be carried through your classifier. So after the classification, so right now in the industry, we only go until this point. We dehull, we mill, we air classify, and we make the fine fraction and the coarse fraction. But there are some research has done, can we further purify just using physical method? I mentioned about a hybrid method, but where you are going to use water in the hybrid method. But this is not a hybrid method. This is just further exploring the possibility of using just physical method of using the electrostatic separation. So when you have the fine and coarse fraction, you will run it through an electrostatic separator. So electrostatic separation is a method to separate particles by using electric forces. It will charge the materials. So electrostatic separation is mainly used in mineral industry. It's not common in food industry, but now it's becoming more common to the food industry too. So triboelectrification is one of the most interesting out of several types of electrostatic charging mechanism for the dry separation. So what happens in this method? This method brings two materials into contact with each other, causing electron transfer from one surface to one material to the other. Various research groups have constructed different electrostatic separators to partition materials into protein-rich and carbohydrate-rich fractions. So it's the same principle. You give the electrons. So based on their electrostatic ability, they will be separated. When it comes to the separation, protein starch mixtures, the protein can be charged to a much higher extent than the carbohydrate, because if you can remember from your A levels or even in your degree, the protein is made out of amino acids, And these amino acids have N terminus and C terminus. So it can be easily take the electrons. So, but conversely, most carbohydrates, they are low ionizable due to the lack of ionizable groups compared to the proteins in the chemical structure. So direct electrostatic separation of starch containing legume flours was shown to be infeasible and need to follow by a classification step, meaning you can't just come to this. You come from seed, dehulling, milling, and you can't jump to electrostatic separation for the separation for protein and starch. First, you have to go through the classification, meaning based on the size and the density, first you separate to find and cause fraction. Only if you want to further purify your material, then only you can use the electrostatic separation. So after protein extraction, what's the next? So we have to modify our proteins because it cannot be used directly in their native forms. Pulse protein ingredients can be incorporated into different food systems to increase the nutritional value and to achieve the desired functionality. Currently, many plant-based alternative proteins include pulses face several formulation challenges, especially in the nutritional, sensory, and functional aspects. Native pulse protein ingredients can contain anti-nutrients, beanie flavor, and in the undesirable functionality. Modification can be done using physical, chemical, and biological methods, or else you can combine all of these to improve the pulse protein ingredients at the micro level of their desired use. These modification processes will thermodynamically change the structure and conformational characteristics of protein. Overall, the modification process expects to improve the protein characteristics. So here you can see there are many methods that you can, after you extract the protein, it can be from wet method or dry method. You have the native protein. So now you are going to use either physical, chemical, or biological methods, or it can be a complex situation like combining all of these. So physical methods are more sustainable, but due to the limited time today, I'm going to only explain the biological methods, the enzymatic method and the fermentation. So in the enzymatic method, a protein can be modified using the control application of enzymes. Like it can be proteolytic or non-proteolytic enzymes. So the desired characteristics can be achieved through the enzyme modification, either by dissociation, meaning you dissociate, you break down the protein, or it can be association, you bring some proteins together, or change the protein structure. So for an example, proteolytic enzymes like pepsin or trypsin, it can be used to hydrolyze peptide linkages in the protein primary amino acid sequence. And then non-proteolytic enzymes like transglutaminase can be used to associate, bring together different proteins and bind them into one sequence through the cross-linking. And the other one is like enzymes like glutaminase can be used to deamidate. Like I mentioned, you can change the structure. Either you break, you bring together, or you cross-link, or either you change the structure. So glutaminase can be used to do that, to change the amide group to a carboxylic group. So then that's the enzymatic method. And then the fermentation, it's a traditional method employed to optimize nutritional properties and the functional properties, and as well as to reduce some off flavors. During the fermentation, microorganisms produce enzymes that will create proteolytic cleavage in the protein, and it will break down into lower molecular weight amino acids and peptides. There can be two different methods of fermentation. It can be solid state and the submerged fermentation. So in the solid state fermentation, you do not require water, or you will only require very limited amount of water. And submerged fermentation requires more water compared to the solid state fermentation. So the submerged fermentation is not preferred. Solid state fermentation is preferred because it will produce higher yield, and it's efficient in waste management, and also it's favorable functional, it's favorable final product characteristics. So after protein extraction and modification, what's next? So now the protein will be incorporated into a food product. Meat alternatives, dairy alternatives, egg alternatives, and other meals and snacks are more popular with this plant protein wave. Due to the limited time, I will be only focusing on the meat alternatives today. So looking at the meat alternatives, first, let's start with what are the ingredients we want to make a meat alternative. So normally, water is the main ingredient, and then it can be texturized base protein, fat, binders, flavor, salt, preservatives, and color. Think like a sausage. For most of the sausages, we use all of these. So instead of meat, we are going to use the texture based protein. It can be from any of the plant sources. It can be pea protein, soy protein, chickpea. So right now, most of you eat TVP. It's made out of the soy protein. So it can be made out of pea protein also. So likewise, so it can be different plant sources to make this textured base protein, but fat, binders, flavors, salt, they are common to all of these. But it's a matter of balancing between the flavor and the other ingredients. So fat can be like either you can use coconut oil, palm oil, or different oil types to match the flavor profile that you need. So when you make the product like a meat alternative, first we need to understand what are the expectations from our consumers. People want to eat meat alternative, which is made out of plant-based proteins, but they are expecting it to act like a meat. They want to eat a sausage that's made out of plants, but they want to act that sausage as of made of animal proteins. So people are expecting certain level of flavor, texture, and appearance. And also we need to consider what the consumer is asking for, whether they are looking for high protein in your meat alternative, are they looking for other iron and vitamins in the product. So we need to be careful, as I mentioned, in the plant proteins, they are lack of some of the essential amino acids and also some of the vitamins. So we need to be careful on that aspect too. And then what types are available? It can be burgers, sausages, nuggets, or either it can be whole cut muscle. Like now there are research done to see whether can we make whole steak or whole chicken instead of making burger patties or instead of making sausage. So we need to consider what's the product type people are looking for. So there are a lot of areas of opportunities because you can change with, you can play with texture, flavor, and the variety. Like right now, there's a market for steak and chicken, and moving forward, people will be asking for goat, lamb, which are made out of plant proteins. And then what's the consumer experience? As I mentioned, most people are expecting your plant-based sausage or plant-based burger patty to act as an animal patty. So when you bite it, whether you can feel that same texture, and when you throw it into a grill in the oven, can you hear that sizzling noise because of the fat from the animal sources? So we need to replicate that with the plant proteins too. So these are the areas of opportunities. We can improve the health by combining different animal proteins to get a complete amino acid profile, or we can combine them to get a higher value of vitamins that are lacking in some of the plant sources. So these are the opportunities that we can play around and make a good product from the plant-based proteins. And the basic method is like you get all the ingredients together, you mix it, and then you form. We call it extrusion. In the next slide, I will be explaining the extrusion. And then either it can be pre-cooked, and then it will be packed and cold storage. So that's the general flow for the making of an alternative meat out of plant-based protein. So this is the flow of the extrusion. So you saw now we have all the ingredients, the plant protein, fat, flavor, and so now we are going to mix it and then form into a meat. So for that, we can do it in two different ways. It can be either dry extrusion, or we call it low moisture extrusion, or it can be high moisture extrusion. So for an example, in Sri Lanka, we normally eat TVP or soya meat. So it comes from the low moisture extrusion, where you can have a lot of air bubbles inside the matrix. In the high moisture extrusion, it's like this. So you don't have that much of air cells in between your structure. So whatever the method, we are going to use extrusion. In the end, if there's a cooling die like this, we call this cooling die, it makes the difference whether it's going to be a TVP or is it going to be a high moisture meat analog like this. So meat alternatives mainly produced through the extrusion method. Extrusion can be either dry like this, or either it can be wet like this. So during extrusion, what happened? We start with the native state. So protein is like this. So protein undergoes the thermal and mechanical stress by heating, pressure, and shearing, which will help to unfold the native globular proteins and form continuous and viscoelastic mass. Because it's important to make this native globular protein to a continuous protein, linear protein, because if you can remember from A-levels or even in your degree, if you are studying the animal proteins, they are more linear, but in the animal proteins, most of the proteins are globular. So if you are going to make an animal alternative product from a plant source, the first thing you have to do, you have to make this globular protein into linear ones like this. So for that, in the extrusion, by using the high temperature, high pressure, and the shearing action, you can make continuous and viscoelastic mass. So this viscoelastic mass can be aligned, cross-linked, reformed, and converted into an expandable structure that has a meat-like structure. So from the cooling die, you can make it more aligned state. So that way it will represent the animal proteins. High moisture and low moisture extrusion methods can be used based on the final product requirement. As I mentioned, do you need a TVP? So you go with this one, low moisture extrusion, and then using the TVP, you blend it with other ingredients, and you can make another burger patty with the TVP. From the high moisture extrusion, you can directly make a meat analog. Like, let's say you go to the market and you buy chicken. So the high moisture meat analogs, idea is to make chicken like meat from one step without going through the low moisture extrusion with blending with other ingredients. So The extrusion process is capable of reducing the heat liable compounds like phytic acid, trypsin inhibitors, and tannin that is present in most of the plant proteins. So it is a very favorable thing to use the extrusion because we use thermal and mechanical stress with the screws. So this also leads to an increase in the protein and starch digestibility and exploitability to produce ready-to-eat food products. Because let's say if you use a plant protein source directly without modification, you might have a lot of anti-nutritional compounds with your proteins. So using this method, you can eliminate most of the anti-nutritional factors because we are using high pressure and high temperature. So now there are many plant-based meat alternative companies around the world, but I would like to take Impossible Foods as an example on how they overcame a specific challenge in meat alternatives. So Impossible Foods CEO and the founder is Pat Brown. He is a medical doctor in his profession. He spent most of his career as a professor at Stanford University. He's well known for his pioneering in gene mapping. However, when he had his sabbatical leaves in 2009, he had a large ambition. So he embarked in his sabbatical leave, 18-month sabbatical leave, and then he had one mission to figure out how to eliminate the meat industry, which he would determine to be the world's most devastating environmental problem. So Mr. Brown ended his sabbatical leave with the conclusion that people would never cut meat out of their diet solely for the sake of environment. Even we saw in the with the Mintel graph I showed you that people are first concerned of their health, not of the environment. Environment and animal welfare comes second. So he concluded that the only way to truly beat meat would be fair and square on the free market. So how would he do that to recreate the meat using plants? Most of their ingredients are similar to what I explained in the common meat alternatives. So today I want to emphasize one key ingredient which makes their product unique. That's heme. So back in 2011, they started with a simple question: what makes meat taste like meat? Then they figured out how to make it with plants. So they found that heme is what makes meat taste like meat. It is an essential molecule found in every living plant and animal, most abundantly in animals, and something we have been eating, craving since dawn of humanity. At Impossible Foods, their plant-based heme is made via fermentation of genetically engineered yeast. They start from extracting the DNA, which makes hemoglobin from soy root nodules. Then they insert it into a modified yeast to make more heme through the fermentation. So if you look at the down left-hand side of the slide, those are the common ingredients. You have the oil and the binders, the proteins and the coconut oil. And this heme is the most important ingredient in their product. So how they extract the heme, they sow in the nodules of soy plants. You know, we have the symbiotic relationship with the legumes and the rhizobium bacteria. So this is an cross-section of the root nodule of soy. So you can see it's in dark red in color. So they figure out what's the DNA responsible to make this leg hemoglobin. So they extract it out of the root nodule, and they put it into the yeast, a special yeast that can reproduce the leg hemoglobin through the fermentation, and they extract that through the fermented method. They extract the heme to be added into their product. I hope this video will work with the sound. If not, it's just whatever I explained in the pictures.
Heme is a beautiful iron-containing molecule, and it's essential for pretty much life on Earth. The heme in your blood grabs the oxygen from your lungs and carries it all around your body. And the amazing thing about heme, besides doing all that sort of stuff that enables life to exist on Earth, is that it's also what makes meat delicious.
We were the first to discover that heme is what makes meat taste like meat. It's highly abundant in animal muscle tissue, and if you were to eat meat raw, that bloody flavor that you get, that's heme. And when you cook meat, with all the different flavors and aromas that you get, that is catalyzed by heme as well.
The craving for meat is really a craving for heme and the iron and protein that it represents in the diet.
Every decision that we make is really driven by our values and our mission. We want to feed the population in 2050. We want to do it in a way that does not destroy the planet. All of the decisions that we've made have been to produce a product that we can make in a way that is scalable and sustainable and safe. And that applies to heme.
We surveyed a lot of proteins and we identified soy-like hemoglobin. Soy-like hemoglobin could come from one of two forms. One would be in its natural form, which is in the root nodules of soy plants. But the other would be fermentation, which is a far more scalable and sustainable way of making that protein. Yeasts are used for making other proteins, certain kinds of beers, wines, and so it's something people are familiar with. The yeast that we use makes its own heme molecule. We take the DNA for the soy-like hemoglobin protein and we insert that DNA into our yeasts.
Heme is what produces the explosion of flavor and aroma when you throw a burger on a grill, and it's what makes the Impossible Burger taste so delicious.
Well, that's the story of Impossible Burger. So I would like to wrap up this session with the research gaps we have seen in the plant protein arena, especially from the protein separation. So there are a lot of opportunities to see what's the most sustainable and effective ways of finding the optimum conditions for protein separation and modification. Like you can combine with can we go with the hybrid methods? Can we reduce more water and energy? Even with the dry fractionation, can we use electrostatic separation? And are there any methods that we can use to further purify our proteins? Or the question comes, do we need to further purify? Because for an example, in a meat alternative, you don't need to achieve like a 90% protein as an ingredient. You can use protein starch blend to make your meat alternative, because that's what we have seen in research, like having higher purity in your ingredient won't make a good final product. Sometimes you need that synergistic effect from the starch, fiber, and proteins to make a good final product. So that's one thing that we need to work on from the research aspect. And then further investigation needs to be done on the G by E, its genetic and environmental effect on the protein separation, and on milling performance, especially how the G by E or the genetic and environment affect the milling performance and their classification, and also breeding programs to improve higher protein values in the our initial plant state, like for an example, pea, chickpea, mung bean, or any other pulse would have like 20 grams per 100 gram, or we call 20% protein in its initial state. So from that 20%, we are extracting protein up to 80% or 90% of protein. So is there any way through the breeding can we improve the plants to make seeds that have like 30% protein, 40% protein? So there are a lot of things to be done in the breeding side as well. And then in some food application, as I mentioned, the purity can be lead to some inefficient raw material and also higher cost. Like when you want to extract higher protein, when you want to have like a higher purity in your protein, it can be costly because now you have to go through the wet extraction, or either you go through dry fractionation and electrostatic separation. So high purity is not, it will not be the always be the answer. So that's one thing to keep in mind. It depends on your what's your final application. First, you have to think, okay, do we need that purity, or are we looking for the functionality? So there's a new concept called now we are not focusing on the purity, we are focusing on the functionality aimed fractionation. We fractionate to a level that we want protein to be act in the final product. Like let's say, even though my fractionation method can extract up to 75%, it doesn't matter, because what's the protein percentage that need to be in the final product? So it will be coming back from the final application. Okay, I'm going to make a meat alternative. So then what's the good protein value I should have? Should I have starch and fiber in my product to make a good meat alternative? So based on that, we will come back and we will decide, okay, this is the fractionation level we need to do to extract proteins. And then finally, especially this is the biggest concern. If you now when we are fractionating, the entire presentation was on the protein. So you might question, so what are we going to do with the other things? I mentioned about dehulling. So with dehulling, you separate most of the insoluble fiber. And then how about the starch? What are we going to do? So there are already started research project on these, valorizing the core products like starch and fiber, how we can utilize those in different food products. So still there's a lot of opportunity to work on this valorizing part of the core products. So then I would like to summarize today's lecture to give you some key take-home messages. So initially, we learned about the need of plant proteins in the current context. There's a growing need because growing population. There's a question how we can feed them with sustainable protein sources. So that's why we need to move from animal proteins, because animal proteins alone can't help this problem. So we need to utilize the plant proteins. And then in the second part, we identified the current, future plant protein sources and how to extract them from the dry fractionation and wet fractionation. And then I talk about how we can modify those native proteins to make them more favorable in applications, like using physical, chemical, biological. I only explained the biological methods, but still you can use the physical, chemical as well. So later we learn about the plant protein products, especially the meat alternative. Other than the meat alternatives, you can go for egg alternatives, dairy alternatives, and the meals and other snacks. So, and then later we learn about the success story from the impossible foods. So with that, I would like to wind up my presentation and open the floor for the discussion and any questions you may have. And again, thank you for inviting me to do this guest lecture, and thank you for everyone who attended today.
Thank you, sir. Now is the time for all of you to ask if you have any questions.
Philip, your mic is not clear.
Hello.
Sir, one has asked a question in chat box.
Okay.
How plant protein are isolated and the novel trends of using plant proteins in biochemical.
How plant proteins are isolated and the novel trends of using plant proteins in biomedical uses. Okay, so I think in middle part of my presentation, I explained the ways that we extract the protein. So I will summarize again the wet fractionation and the dry fractionation. Wet fractionation is costly and it also uses the chemicals and the water, and it's not sustainable at this point, but you can achieve a high purity. But for most of the soy proteins, that's how we do. But the dry fractionation is more sustainable. We only use air. We mill the materials into small particles, and then we separate it based on the size and the density. So those are the two methods. Moving forward, what we can do, we can combine these two. We do the dry fractionation and put another layer of wet fractionation, not using chemicals, but when we want to further separate the proteins out of starch, we can dissolve them in water and we can separate the protein and starch further. So that's the summary of how we can separate the protein and starch, or how we can isolate. So the other thing I wanted to clear, by the wet fractionation, when we get the protein, we call it protein isolate. When we separate protein from the dry fractionation, we call it protein concentrate. So that's the main difference. And for the biomedical uses, I am not an expert in the biomedical field, but my guess is for most of the cosmetic products, I have seen a lot of research has been done with using the starch. Especially, I was telling like what can we do for the co-products that we separate from the protein, how we can use that fiber and starch. So especially for starch, it has been used in a lot of tablet fillings, in the biomedical field. For the filling for the medicine, you need to have a filler. So that starch can be used in the like the plant protein starch can be used in these fillers. And also in the cosmetic side, you can use the starches to make your makeup or whatever the cosmetic that you are using. So I hope that answered that question. So I see another question, so I will go ahead and read it. Protein is the main component of feed ration calculation in cattle management. Can you explain how it can be obtained cheaply from plant and how to calculate it best way to high productivity? How is the field of nutrition management? Okay, as far as I understand, I think it's asked how we can cheaply make the plant proteins, because yes, I think I can't see who asked the question, but the person who asked, I think he's referring to the cattle management. So normally for the animals, we feed them the lowest quality materials. So the same with the plant proteins, what we can do if we want to make cheaply the protein cheaply for the animal feeding, the thing is you can use the rejects. For an example, so in chickpea, we have the whole chickpea, and when you handle it, it will be split, so it will be separated. So in human food consumption, it is having a low value. Like if you want to, so think of it as this. If you are a farmer and if you want to sell your chickpea to a company who is buying chickpea, if you can sell the whole grain, you can get a premium, like you can get high value for your product. But while you are harvesting your chickpea, if the chickpea is splitted, when I say splitted, it's like two parts and or broken, so then you will have a lesser value from the market. So it's considered as a damaged product. So if you want to make cheap protein for the animal feeding, what you can use, what you can do is you can use the that damaged or broken seeds, and then you can follow the same procedure, but not going to the wet extraction method. You go for the dry fractionation method, which is cheap, very cheap compared to the wet extraction. You don't use water, you don't need any chemicals. You just need a good milling system to mill into the certain particle size, and a good classification system to separate them based on the size and the density. And the other thing I want to mention, so this whole idea of plant protein is to get away from the animal protein. So for me, it won't make any sense to make plant proteins and again to feed the animals and get the animal proteins again back to our human consumption. Because as I mentioned, animals are very inefficient in protein conversion. When you, I can't remember the values on top of my head, but when you feed and chicken to get a 1 kilogram of chicken protein out of chicken, you have to feed, I don't know, maybe 50 times of protein to that animal. So it won't be sustainable in the bigger picture. But as for now, the cheapest way will be use the rejects from the human consumption, but still you have to select the quality material, because you have to test for aflatoxin, you have to test for other quality parameters. Then you can go with the dry fractionation, make the protein, and then based on the cattle feed requirement, you can make the products to cattle feeds. One question again, can you explain the research areas related in plant proteins? I think so. The entire presentation covered all the research areas we have been doing, like a research community. Every other research team has been doing. So for an example, so I mentioned the research gaps. So those are the areas that you can work on if you want to further study this area, like starting from the field, like how we can increase if you are more into breeding side, how we can increase the protein, or how we can reduce the anti-nutritional factors in the seeds in the plant level. So then from the plant, then you move to the ingredient, like the protein, starch, and fiber. So now you can work on how we can more efficiently separate the starch and protein, rather than the method that now I explained. I mentioned there can be hybrid methods, or there can be some method similar to the electrostatic separation. So electrostatic separation is somewhat new method people have been start using. It's not commonly used because it's expensive at the moment. So there's a lot of opportunities to work on that area too. And the other thing, after you extract, so that is extraction, and then how to modify. I showed a graph saying these are the methods, like physical, chemical, and biological methods. And even in this physical method, it can be heat, it can be sound, it can be ultrasonic, or whatever the physical method, you can work on that area, like how we can optimize. For an example, let's say if we have a problem using the chickpea or pea, especially with the pea protein, it's not that favorable. Like you have that beanie taste, like you when you taste the pea protein, you taste it like cardboard, some people call it. Some people call it muddy, or some say beanie. So you can use heat treatments to reduce this beanie flavor. So now the problem is, now the research area opens, okay, what's the temperature, what's the time, what's the machine we have to use. So those are the things that we can work on to identify what's the optimum condition to make or modify the plant protein to make it more favorable. So that's only one example. You can think of many different things, like even just going back to the slide I shared you, different modification method. Even within different modification method, what are the optimum conditions? So that's up to the modification. So then we come to the actual product development. So now we have the modified protein. So let's say we are going to make a meat alternative. So what is the correct ingredient formulation? How much protein, how much fat, how much starch, how much flavor we need to add, and what's the way how we can do the extrusion, and do we need to go with the dry extrusion, do we need to go with the wet extrusion, and what are the optimum conditions? Because I didn't go into deep details with the extrusion. Extruder is like, it has different screws. It can be twin screw, single screw, and you can change the speed, you can change the rotation, the way that it rotates, and the temperature within the barrels. So there are a lot of opportunities in that area too. And then after you get that product, let's say you make a meat alternative, now you have to feed the people. Now you can come to the sensory testing. So in that sensory research area, you can work on, okay, what are the new modifications we have to do? So from the plant breeding to presenting it to the consumers, have a lot of things to be done. So those are the research areas you can work on. What do you think plant protein is more useful than animal protein? Can you tell us the particular name of plant protein? So I believe, if I understand it correctly, so animal proteins, as I mentioned, the problem with the animal protein is, let's say if you are going to eat chicken or beef or pork, you are not only eating proteins, you are going to eat the fat. Or so the main problem is animal protein contain cholesterol, but plant protein, it doesn't have cholesterol, so it's more healthier compared to the animal proteins. And then the other problem is, as I mentioned, the environment and then the animal welfare. So if you care about the environment, it's time to move from the animal or being eating just meat to move to a vegetarian or vegan. But I don't suggest you 100% convert it to a vegetarian or vegan. If you can at least be a flexitarian, as I put it in my poll. I know it didn't work with our technical difficulties, but still, what I wanted to see how many people have been eating as vegetarians and vegan and the flexitarian. So flexitarians means whoever eat both meat, animal meat and plant-based products. So in US and the European countries, most people are now converting to become more flexitarians. So that way you still enjoy the animal meat. And especially in Western countries, it's really hard to change people's mind to become just to eat plant protein. So it's a matter of how we can slowly convert people into eating plant protein, because the bigger thing is, okay, think the good example is Sri Lanka. How many people, our population is keep increasing. How we are going to feed our population? So, you know, there's a lot of problems with our agriculture system, even in Sri Lanka. Like we have a very limited amount of land, limited amount of water. So how we are going to use this land and water to make more agricultural product in terms of like proteins to feed the ever-growing population? Like we don't have much land. Like in US, the state that I'm living is like 7 times bigger than the Sri Lanka itself. So when you go out, you can see a lot of fields. It's basically an agricultural state. So Sri Lanka is going to face a bigger problem in terms of how we are going to feed this ever-growing population. So it's a matter of we are changing our habits, changing from animal protein to plant proteins. It's only one thing for Sri Lanka, but in the world is the biggest thing, because here we have enough land, enough water, but still we have to find more sustainable ways how we can feed the people. And the other thing, since it is inhumane for how animals treated, like in segregated areas, as I mentioned, most of the diseases, like even, I don't know, this might be not 100% correct, but most of the pandemics started with the animal-based sources. It came from animals to humans. So those three are the major things that is important to move from animal proteins to plant proteins. And the only downside of the plant protein is a single plant protein is not a perfect protein, I would say. Like if you eat a pea protein, you can't get all the amino acids. It can be lesser in sulfur-containing amino acid, like methionine and cysteine. So the good thing is, like, we can mix and match. Like we eat in Sri Lanka, we eat rice and lentils together. So that means cereals like rice, it lower in lysine, and pulses like lentils, it's lower in methionine and cysteine. So we match them together, we eat them together, so we can get a complete essential amino acid profile for our proteins. I hope I answered that question. So the next question is how much we should include plant proteins in our diet. So it depends. Like here, there's a daily value on the labels. Like there's a in US, I can only speak of US since I have an experience on the labeling. So if you have a governing body that dictate, okay, at least you have to eat 110 grams per day from 110 grams of protein for your entire meal. So it depends on the calculation and how I would say it depends on person to person, but at least in general, it depends on from country to country how they govern or how they regulate, saying, okay, eat this much of protein per day. So as I'm not an expert in the nutrition science, I don't know the exact value, how much to eat per day. But I think you can see by yourself, like saying, okay, even right now, we need to calculate ourselves, especially with the PDCAS values. We need to consider, even though we are going to eat a lot of proteins, whether is it going to be absorbed into our body. So there are many things that you need to consider. To be honest, I can't remember the numbers, how much you need to eat proteins in our diet, because it depends on person to person. Because sometimes I may be older than everyone else, and I might have a different protein requirement, and also I might be suffering from some illness, like let's say I'm immunocompromised, so then I might need to eat some higher protein values than to others. So it depends on person to person, so it's really hard to say a value at this point. Another question, any possibility to alternate wheat 100% from another cereal? The problem with, I think, as far as I understand, so the question tends to ask like, can we replace wheat from another cereal? The question, the answer will be, the simple answer will be no, because you can't match rice flour with wheat flour. The main problem is, in the wheat flour you have the gluten, in rice you don't. So in there are a few different gluten containing cereals like wheat, rye, barley. So still, even within the rye, barley, and wheat, there are differences in the protein the protein percentages. Like it is not only the gluten, there are different other proteins who contribute to make the wheat flour, and also the starches and the fibers. So it won't be possible. I mean, it will be possible if you mix rice with gluten that is extracted, but you can't find a alternative crop to the wheat. So wheat is unique, wheat is wheat, rice is rice. So if you want to make a gluten-free product, that means you need to have some starches, binders, so your ingredient list will be increased. So let's say you are going to make a gluten-free rice bread. So how are you going to give the structure to that bread? Like a wheat, like normal day-to-day life, we eat the bread, a good structured bread. So how are we going to give that structure? Because as a consumer, I don't like to see like a flat bread, like a roti. I don't need to call that roti bread. Sometimes when people make rice flour bread, it's like a roti. I mean, there's a lot of a very small volume. So gluten gives the structure. So if you are going to replace the gluten, then you need to find different alternatives like binders, hydrocolloids to give that structure. So that's my answer. So you can't replace a plant 100%, but for the formulation standpoint, you can do some manipulations with other ingredients like starches and hydrocolloids. Okay, so the next question is what are the future trends of plant protein? So I briefly touched this area on the protein sources. So we have established and novel and future. So established, current, and future. So established proteins, mostly like animal proteins, and then all the animal proteins like egg, meat, and then milk. So then the established plant proteins are like gluten, and then soy protein, pea protein. So those are more established markets, established plant proteins. And then the current ones are now the improving ones are mostly into the chickpea and then the bean proteins. And then as for this question, for the future trend is in looking into some seaweed protein and then potato protein. So those are in terms of plant-based. And then for the animal-based future trends, looking at some insect proteins and like larvae, crickets. So how can we extract those? Already in US, they have started large factories to make larvae protein, like we call Salabeage panua, Evelyn protein. Then other than that, the crickets, you can grow crickets like the Tanakolopetta. You can grow crickets and then make the powder, protein powder out of the crickets. And the other thing, the increasing trend is to have fermented products. I briefly touched even with the modification. So with the fermentation, you can make whole muscle meat alternatives, like, because the fungi can give the structure, the mycelia can give the structure. So the biggest challenge with the whole muscle cut protein, whole muscle cut meat from the plant-based protein, like when I go to an restaurant, I would like to eat a whole chicken or whole steak, we call it. So how we can make that steak, like think of as when you go to the market, you get that pieces of chicken. So how we can make that chicken pieces out of plant protein? So for that, we can use the fermentation, because it gives the structure for with that mycelia. The other question is chronic diseases focus food supplement that could be made using plant proteins. There can be many problems. As I mentioned right now, there are some deficiencies in the nutritional profile using single protein. But compared to animal proteins, it is not leading to that much of chronic diseases. But there are problems, not chronic diseases as in animal proteins. Can we use the plant protein for the dairy industry as the alternative for cow milk because flavor texture will be changed? It's a good question. So right now, in one slide I mentioned you can make milk alternatives and dairy alternatives. So that's the dairy or the cheese made out of plant protein. So the basic thing is you get the protein, plant protein, and then you use the fermentation. Like for an example, but the thing is you have to use a specific strain of yeast to do the fermentation to make cheese out of plant protein. Yes, for the question, the answer is yes. Flavor, texture will be changed. That's a research area that's a good opportunity to work on. So still there are research going on. So there's no right answer right now. Okay, you use this plant protein, use this yeast, ferment for this time of period, and you can get the 100% replication of the cheese that you made out of animal milk. No, there's no such an answer. So it can be different from one process to another, and also like it might be different. My taste might be different from the others. So yes, the flavor and texture will be changed, but you can do modifications to at least achieve like a 90% replication of the animal dairy. Yes, I will share my email here. I think I answered all the questions I see in the chat. If any questions anyone needs to ask, this is the time, so it's open.
Thank you, sir. Are there any other questions?
So anyone who is interested, I just typed my email. So if anyone wants to reach out, you are more than welcome. I'll be happy to help. And if anyone wants to learn more on this topic, I have my own 2 review papers I recently published. So I will be happy to share that as well.
Time goes by. I think the questions are over. All are clearly identified, haven't any doubt, and more helps to improve our knowledge. Now I cordially invite Senior Lecturer, Head of the Department, Mr. Kamal Edurisinghe, to present an idea.
Dear Dr. Supun, Fernando, very good morning. Not of course you are in country in the morning. And it is a great pleasure to us and sharing your ideas and experiences with our students and young bloodies. And also that is a good eye-opening session for our students and some staff members of that the new technological era, how are going to or any alternatives related in the plant using in the plant protein. That is a great session and very informative, and that is a very vibrant session. So I am very much appreciated to you in the commitment and the valuable time sharing with us and within the Sri Lankan. So the new technological development era of our country and current status are converting and focusing in education also in some technological development. That is the trends coming on in our education system. You are the pioneer person in serial sciences in the field of that. In the globally, you are disseminating your knowledge and our nation. That is the situation that we are expecting. You are sharing in future knowledge with us and coordinating with our department for further collaborative research. And this is the very bad situation currently we are facing due to the COVID situation. But we are grabbing and overcoming for some problem and solutions are finding. Thank you very much, Dr. Supun, and as the product of USJP. There are I also in the USJP in past association, and Dr. Randhya also attend to this session. I know that good for well. Thank you so much. Then we will get together, work and collaborative research. I expect to work together. Thank you very much. Again,
Thank you. So thank you for inviting me to give this session. So yeah, as mentioned, Erandhya was my colleague and we were friends. So I'm happy to see her here too. So even in future, if there's anything I can help with, any lecture I can give, I will be happy to help because I want to disseminate this knowledge. As you mentioned, I want to disseminate this knowledge, whatever I'm learning from here in US, because I know sometimes because of the technological problems and even with the COVID, there are a lot of challenges in Sri Lanka. So I'll be happy to help to transform that knowledge, whatever I learn from here, to Sri Lankan students, so they can pursue their careers, and even to help them to further go in their studies, to go for a master's or PhD in their career. So I'll be happy to help in future. Thank you.
Thank you, sir. Now I invite Secretary of Food Technology Association to deliver a vote of thanks.
Thank you, Maheshi. Honorable guest speaker, Mr. Supun, Sandaru Fernando, respected our HOD, Mr. Kamal Edrisinghe, and all the members. It's such an honor for me to get the opportunity to thank you all on behalf of Food Technology Association. First of all, I'm really sorry, sir, the technical errors that occurred at the beginning of the session. I extended my gratitude to our honorable guest speaker, Mr. Supun Sandaru Fernando, to take out time from his busy schedule to this base event. And thank you for giving us knowledge, inspiring and encouraging us with your word on this special event. And special thanks to Mr. Kamal Edrisinghe, our HOD, for providing support to make the webinar session successful. And also I would like to thank Ms. Hasini Vijaytilaka for inviting sir to conduct this lecture. I think I must thank organizing team, volunteers for working hard in the past few days to make this event successful. Finally, I will thank Vice President of Food Technology Association and all other office bearers, and also members present here. Once again, thank you all of you on behalf of Food Technology Association. Thank you. I'm Buddhini Udeshika.
Thank you, Buddhini. Thus concludes our third guest lecture with great success. Thank you to all who attended. All are stay safe and very carefully while following proper health practices as the COVID-19 virus is spreading very fast. Thank you, sir. Thank you all.
Thank you.
Thank you, sir.
Thank you, sir.